Okay, today we're talking about the autonomic nervous system, and we're going to start by mentioning the cranial nerves, which supply innervation to the head region. And for those cranial nerves with a motor function, we saw, for instance, that your eye muscles were controlled by cranial nerves 3, 4, and 6. And your facial muscle was controlled by cranial nerve 7, and so on. The muscles that are controlled were all skeletal muscles, which we could move at will. So that was the somatic pathways. Then there were those cranial nerves that you had to know visceral motor function for. And so today, that's really what we're going to be talking about in general, visceral motor pathways. And I want to stress the motor aspect of that. There are certainly visceral sensory pathways. Sensory neurons located in the dorsal root ganglia receive information from the interoceptors within visceral targets. Sensory information about temperature, chemical changes, irritation within the visceral organs, like stomach pain, can be felt consciously but is often processed and dealt with at an unconscious level. But otherwise, the visceral afferent pathways are similar enough to the somatic afferent pathways. On the other hand, the efferent division, we could already see here, is getting a little more complex. The efferent pathway is split into motor neurons. One, the somatic motor neurons, which innervate skeletal muscles, which we have so far focused on. The other, the visceral motor neurons, innervate visceral targets, namely smooth muscle, cardiac muscle, and glands. These visceral motor neurons belong to the autonomic nervous system, which is split into two divisions, the parasympathetic and sympathetic. So we'll be comparing here the somatic and visceral motor pathways first, their regulation, targets, and different pathways. And then within the autonomic nervous system, we'll be looking at those two pathways that also differ both structurally and functionally. So back to my favorite nervous system picture, we talked about how commands from the primary motor cortex of the cerebrum issued descending commands which travel down the spinal cord, out the spinal nerves into its targets. So we associate descending pathways, efferent pathways, and motor together. And then we add in the cerebral cortex and skeletal muscle to signify that it's a somatic pathway. The visceral motor pathway is controlled by a different part of the brain, so commands are issued down descending pathways, out spinal nerves, and the efferent motor pathway to innervate smooth muscles and glands. So from here, the beginning of the pathway and the end are different, and as we'll see, there are further differences in between. So we're going to first start looking at the difference between somatic and visceral targets. Here, the ocular motor nerve contains somatic motor neurons controlling the eye muscles, which will allow you to voluntarily move your eyeballs up and down and sideways. So when your teacher tells a bad joke, you can voluntarily restrain yourself from rolling your eyes upward or not. The ocular motor nerve also innervates the smooth muscle within your eye that controls pupil constriction. So here, if a bright light is shined into your eye, the smooth muscles will close up automatically. You can't voluntarily do this or stop from doing it. There's also muscles that automatically bend your lens to allow them to focus whatever you want to focus on. This again is as involuntary as when you are taking a picture with your iPhone and watch it automatically focus up and down on the center object. So the bottom line is there's two examples of smooth muscle in their operation. Usually the smooth muscle we'll actually be talking about is smooth muscle that surrounds your gut tube or blood vessels. The other major visceral target is glands. In this case, the stimulation causes secretion of whatever that gland makes. So cranial nerve number 7 is linked with your salivary glands, and stimulation there causes secretion of saliva in your mouth. Again, that is obviously involuntary. It occurs when you smell food, see food, or even think about food when you're hungry. There are also all the glands within your digestive system that secrete things like digestive juices. So there's just a few examples of where you'll find smooth muscle as well as glands, and there's also cardiac muscle and a few other effectors for your visceral effectors. So the next major difference between somatic and visceral motor pathways, as we've already suggested, is control over this pathway, and that is what part of the brain is the master regulator. For somatic pathway, you've already known that the primary motor cortex is the final output for motor commands that will ultimately control the movement of your skeletal muscles. Mind you, not every single muscle contraction you make is voluntary. Much of it goes on at a subconscious level, but most of it can be voluntarily controlled. For your autonomic system, we're kind of on autopilot. Your autonomic system is controlled by the hypothalamus in conjunction with the brainstem, but we're going to consider the hypothalamus as the master regulator because the hypothalamus will ultimately control the brainstem too. The hypothalamus does a lot of things, but we see here a couple of nuclei that are groups of neurons that are responsible for controlling the output of the sympathetic and parasympathetic nervous system. Now in reality, there's no such thing as a master regulator in either system. They're all working in conjunction with extensive crosstalk within the two systems. This is why you can think of a delicious food and your mouth will water, or why your emotional or hormone-soaked brain will short-circuit your rational faculties. So command center hypothalamus targets smooth muscle, and the last difference between the somatic and visceral motor division concerns the peripheral efferent pathway. That is, what happens as the visceral motor neurons in the brain stem or spinal cord travel out toward its peripheral target. So there's sets of visceral motor neurons within the lateral horn and parts of the spinal cord, as well as nuclei in the brainstem. They send their axons out the same pathway as the somatic motor neuron, so your spinal cord visceral motor neurons' axons leave through the ventral root. So let's go back to the somatic motor neurons of the oculomotor nuclei controlling the eye muscles. Axons leave the brainstem, branch off to connect their axon terminals to the skeletal muscle fibers at the neuromuscular junction. Axons of visceral motor neurons exit the brainstem toward the peripheral target, but do not innervate that target directly. Instead, they enter a ganglia, or more specifically, an autonomic ganglia. Remember, a ganglion is a group of neural cell bodies outside the central nervous system. So the visceral motor neuron, whose cell body is located in the brainstem, sends out its axon, and that's considered the pre-ganglionic neuron. Its synapses within that autonomic ganglia with a post-ganglionic neuron that resides in that ganglion. That post-ganglionic neuron receives the message and sends it to the final target. In this case, that might be the ciliary muscles that help focus the eye. But this will go for all those cranial nerves with visceral functions, as well as all the autonomic nervous system. There's groups of these post-ganglionic neurons and autonomic ganglia all over your face and body. So these constitute the second set of ganglia we have encountered, the other one being the dorsal root ganglia, or similar groups of ganglia in the cranial nerves. So this is your general organization of the autonomic nervous system. A centrally located neuron in the brainstem or spinal cord sending its pre-ganglionic axons out to synapse with a post-ganglionic neuron within an autonomic ganglia. The post-ganglionic axon then relays that message to the visceral effector organ, whatever that may be. So with all those cranial nerves, whose cell body resides in the medulla and the pons and the brainstem, they are connecting with those post-ganglionic neurons in an autonomic ganglia, not the target directly. So that second set of peripheral neurons is the last major difference between the somatic and the visceral pathways, and also marks a major difference between the two divisions within the autonomic nervous systems, as we will see. These two divisions of the autonomic nervous systems are most commonly referred to as sympathetic and parasympathetic divisions. Those names don't really give you any clue as to their location or their function or anything else, but there are differences between the location of the nuclei, the location of the autonomic ganglia in relation to their target, and most famously to their function. So we're leaving the somatic nervous system to talk solely about the two divisions of the autonomic nervous system. Anatomically speaking, the first thing we can do is look at the location of the visceral motor neurons, those pre-ganglionic neurons within the central nervous system. Those four cranial nerves, which make up the bulk of the parasympathetic nervous system, all reside within the cranium. The rest all reside within the sacral region of the spinal cord. So it may be best to remember the parasympathetic division as the cranial sacral division. And here I'm going to let you guess where the visceral motor nuclei of the sympathetic division or thoracolumbar division reside. So the craniosacral and thoracolumbar division are called for the names of their pre-ganglionic cell bodies within the central nervous system. So that's the first main anatomical distinction between the two. So let's look first at the craniosacral or parasympathetic division. So the craniosacral division is located in the cranium and the sacral region of the spinal cord. Its function is generally characterized by the phrase rest and digest, or feed and breed. This relates to its role as operating as normal background to maintain homeostasis and to conserve energy when we don't need to expend it. So in relation to the feed of the feed and breed, it can be shown in its effects on the salivary glands. As mentioned, when you're hungry, it begins to get your body ready to digest food. Not everything about these functions are going to fit nicely in a nice clean box, but in general, the activation of the parasympathetic pathway increases the activity of your digestive system while decreasing the activity of things like your heart rate as well as your respiration rate. Much of that is due to cranial nerve number 10, the vagus nerve, which I'll get into in a second. Those sacral division have complex output to the urinary and reproductive system. That breed part reflects the fact that blood flow toward the external reproductive organs is increased with parasympathetic input. But back to cranial nerve number 10, the vagus nerve. This is the major output of the parasympathetic nervous system. It has functions, but this part of it dangles down into the thoracic and abdominal cavity to innervate the heart, the lungs, the liver, the stomach, pancreas, etc. And again, we're generally increasing digestive functions and decreasing activity in systems like the heart rate and respiration. I'll mention that in the parasympathetic division, there is no innervation of the smooth muscles of blood vessels, so it does not generally control systemic or regional blood flow like we'll see with the sympathetic nervous system. So here's a close-up of some of those organs, and we could see the preganglionic fibers, those axons that have left the brainstem and headed out toward the targets. These preganglionic fibers, in red, nearly reach their targets before entering that autonomic ganglia. Those autonomic ganglia for your parasympathetic targets are very close to or even embedded within the targets themselves. So when comparing the parasympathetic and sympathetic pathway, that's one thing you're going to want to remember. Your autonomic ganglia is embedded within or close to the effector organ. Once you know that fact, you can deduce the preganglionic fibers are relatively long and the postganglionic fibers are relatively short. So your vagus nerve is leaving your brainstem, and its axons travel down a distance to reach the postganglionic neurons in the parasympathetic ganglia close to the target. So we're going to compare that with the sympathetic ganglia, which are located some distance from their target. So this brings us to the sympathetic or thoracolumbar division, where the cell bodies of the pre-ganglionic neurons are located in the lateral horns of the spinal cord in between T1 and L2. The sympathetic division function is commonly characterized with the buzzwords fight or flight. Really, the sympathetic division kicks in whenever you are needing extra oxygen, more blood circulation to the muscles, and in which case you don't need to be digesting food, so sympathetic activation slows down that whole digestive system. This means, among other things, your mouth might go dry because salivary gland activity has decreased, and movement of food through the digestive tract is also slowed down. And the ability to direct oxygen-rich blood away from your digestive system towards your muscles is also controlled by the sympathetic's innervation of the smooth muscle lining blood vessels, which is also, again, unique to the sympathetic division. As far as sympathetic innervation to the reproductive organs, I'll let you figure out how this fits into fight or flight, but the sympathetic activation is responsible for, mechanically speaking, orgasm and ejaculation. Or, as some people put it, the parasympathetic points and the sympathetic division shoots. So we see here that the sympathetic and parasympathetic targets are often the same, although there are some important differences, and their actions are generally opposed to each other. In reality, it is not that you have your sympathetic or parasympathetic division on or off. There's a constant balance of activity between the two, regulating your visceral response and activity. One unique target of the sympathetic nervous system is the adrenal medulla. Stimulation of the adrenal medulla causes epinephrine to be released into the bloodstream, where it's going to have widespread effects that you are likely familiar with things like adrenaline rush. The heart rate increases, you're highly energized, your blood flow to your muscles increase. So that's stimulation of a single target that has widespread effects all over your body. Also, the effect will last longer than the initial stimulation warranted. When your heart rate increases due to direct stimulation of a sympathetic nerve, you can stop that as soon as the impulse stops. However, with adrenaline, it is still floating around in your blood for a lot longer. That's why if you're scared or angry, and if you have adrenaline rushing, you can remain in an excited state even when everything else tells you to calm down. So again, this is unique to the sympathetic nervous system. The adrenal medulla has no input from the parasympathetic division. So those are the functional differences between the sympathetic and parasympathetic. And to recap, your parasympathetic nervous system has those parasympathetic ganglia embedded within or close to the organ, which meant long preganglionic fibers and short postganglionic fibers. For your sympathetic nervous system, the preganglionic motor neurons in the thoracic lumbar region, residing in the lateral horn of the spinal cord, send their axons out the ventral root, and those axons usually synapse with the postganglionic neurons in the sympathetic ganglia that live right alongside the spinal cord. So the sympathetic ganglia are pretty close, which means they're far away from the target organ, opposite to what we see in the parasympathetic ganglia. Those ganglia are linked in a particular way and so are referred to as the sympathetic chain ganglia. So that's the name for the paired structures running laterally alongside the spinal cord, although there's also unpaired ganglia lying anterior to the spinal cord. Those ganglia go on to innervate the unpaired viscera, the abdominal cavity. We'll keep it simple and just concentrate on the sympathetic chain ganglia. The significance of the length of the preganglionic fibers and the postganglionic fibers become more evident as you learn about how each of these respective fibers are myelinated, which is going to affect the speed of conduction along the length of those fibers. You're going to have to wait for physiology to learn about that, but maybe you can keep the anatomy in mind when you do. Also, in a related way, there is one target of the sympathetic nervous system that does have a very long preganglionic fiber that reaches its target directly. The target is the adrenal medulla, a major factor in the sympathetic response. So to summarize the differences between the parasympathetic and sympathetic anatomy, your parasympathetic nervous system with those visceral motor nuclei residing in the cranial and sacral regions of the central nervous system has those parasympathetic ganglia embedded within or close to the organ, whereas the sympathetic division has the motor nuclei in the lateral horn of the spinal cord's thoracic lumbar region, has those sympathetic ganglia close to the spinal cord. Functionally, we saw the parasympathetic division involved in normal homeostasis, maintaining low energy and increased digestive activity, whereas the sympathetic division prepared the body for more vigorous activity. There's usually a balance between the two, and they constantly adjust to meet the needs of any given situation. So we want to compare those two divisions of the autonomic nervous system, and we want to compare the autonomic system in general to the somatic motor system. And that's all you got to know. See you next time.